Hi everyone, I'm Elisa Hathaway, and today I'm going to be presenting on evolutionary evidence, developmental mechanisms conserved between invertebrates and vertebrates. It is crazy to think that worms and koalas and humans all share some distant ancestor. I mean, we don't even look remotely alike, so how could this be? Well, it's what's inside that really matters. By comparing invertebrates and vertebrates, there's actually so many similarities across neural development. For example, long-term memory across species such as in sea slugs, fruit flies, and mice is possible due to cyclic adenosine monophosphate, or CAMP-responsive element binding proteins. Another example is apoptosis, or programmable cell death. Cell death sounds scary, but it's actually very helpful in some situations, such as in fruit flies for dorsal closure, for deleting structures, such as the tail of a tadpole, or for sculpting, such as in human finger forming. In humans, without apoptosis, cells can keep dividing, leading to cancer. Further, neurons throughout evolution have many similar features. Most neurons are electrically excitable and have long processes. Both hydras, which are invertebrate freshwater animals, and humans have voltage-gated ion channels. Let's spend some more time talking about specific comparisons by first discussing the embryo similarities between vertebrates and invertebrates. Similar molecules pattern the dorsal ventral axis. The Drosophila embryo resembles an inverted xenopus embryo with an ectoderm and mesoderm region. The transcription factors that pattern the embryo have similar functions. For example, the equivalent of SOG in invertebrates is cordon in vertebrates, and they both play a very important role in dorsal ventral patterning, which establishes various ectodermal fates. The dorsal lip in vertebrates acts as a neural inducer and is the overall organizer of the entire body axis. The Spearman-Mangold experiments showed that the dorsal lip could produce a whole new body, such as this newt. In invertebrates, cordon mesoderm has a similar function and induces the neural system development. The mesoderm in both vertebrates and invertebrates gives rise to the notochord from cells that become rigid. The formation of both a notochord and a neural tube is a prerequisite for the development of a bilaterally symmetrical body characteristic to all chordates, including lancelets, tunicates, and humans. If you're unsure of what a notochord is, I talk about it in my previous video. These are just a few examples, but looking at similarities in neural development across species provides hundreds of clues for evolution. Another great example of similarity across various species can be observed by looking at neural development in Hox genes. Hox genes are a subset of homeobox genes, which are transcription factors that use homeodomain to bind DNA and regulate transcription. Hox genes are different because they can transform body parts. They are responsible for determining the general body plan from the number of body segments to animal head-tail directionality. The Hox gene clusters have a similar anterior-posterior spatial organization along chromosomes called collinearity. In the Drosophila Hox gene cluster, the anterior-most expressed gene is 3' and the posterior-most gene is 5'. For mice and humans, there are 4 separate Hox gene clusters on 4 different chromosomes, but the order of gene expression is still in a 3 to 5' order. Genes that are duplicated within an organism, such as A1, B1, and D1 in this case, are called paralogs. Genes similar between species, such as IAB, A1, B1, and D1, are called orthologs. Vertebrates segment more posteriorly during progressive growth, so Hox genes are activated in a timed sequence which follows their 3 to 5 chromosome order. This is referred to as temporal collinearity. Posterior induction is likely to be the basis of temporal collinearity. The most 3' anterior Hox genes are expressed earliest, and they in turn are proposed to actively promote the expression of the next more 5' posterior genes, and so on, sequentially. For example, it would be A1, then B1, then D1, and then A2, B2, etc. The base Hox gene is similar, but humans and mice have more, so an error doesn't make that much of a difference. But the elimination of the Hox gene cluster in Tribolium beetles results in all segments having identical morphology. Image A shows a normal beetle, while image B shows a beetle without the Hox gene cluster. The normal number of segments develop, but all segments look like the antenna segment. This shows that the Hox gene is extremely important for positional identity. It would be very unfortunate if just missing one Hox gene cluster resulted in such a significant effect on humans, such as just having a bunch of arms. Thanks to evolution, this isn't the case, since we have a lot more backups. What do I mean? Well, we can think about our gene expression as the Russian dolls, Matryoshka. The expression of our genes are overlapping, so the deletion of one doesn't result in completely missing structures, and the error is thus less damaging. In mice, the expression areas of the genes in head brain are overlapping from OTX2, OTX1, EMX2, and EMX1. Due to posterior prevalence, as mentioned earlier, if all paralogs are knocked out, the more anterior head structures develop. Thanks to evolution, basic mechanisms that existed in invertebrates have developed and improved so that more complex species like humans can exist. There are so many developmental similarities across organisms, indicating that all of us are linked in some way across time. Thank you for your time and for listening.